Okay, so our research is on the prokaryotic genome assembly and annotation of abandoned acid mine drainage. My name is Abby Cunningham.
I'm Christine Burey.
And then our faculty sponsor was Dr. Volkanis. Right here, we have a picture of the water in Boyce Park. So abandoned mine drainage is a common form of water pollution in areas where mining has taken place in the past. It is polluting the world and specifically our drinking water and agriculture. These mines occupy over 500,000 miles of land in the country, and they also lead to heavy metal contamination in rivers, lakes, streams, and other natural habitats. Many bodies of water that are impacted by abandoned mine drainage have pH levels of 4 or lower, which is similar to battery acid. This drainage forms as a result of a chemical reaction between water and rocks that are containing sulfur-bearing minerals. Through testing the contents of abandoned acid mine drainage, we are able to better identify its components so that in the future we can further test them in order to create a treatment system. Boyce Park is located in Pittsburgh, Pennsylvania, and has unfortunately been exposed to acid mine drainage. We hypothesized that if we could test the water from Boyce Park and identify its components, then we could use our data that we received to potentially in the future design a treatment plan that could purify and treat contaminated water.
Right, so I was talking about materials and methods. So our first method we used was serial dilution. We separated out bacteria from the culture that was given to us from Boyce Park, and we got the colonies we got them onto R2A auger plates. The next thing we did was we did streak plates. We used confluent growth that occurred on R2A plates, but as the gradient lessens, single cells will then be deposited, will separate from other cells. These cells will give rise to individual colonies. Then we picked these colonies with inoculating loops and transferred them into new media for maintenance and purifying of a culture. The next thing we did was we extracted DNA. This is a method of purifying DNA by using physical or chemical methods from a sample separating DNA from cell membranes, proteins, and other cellular components. So we just wanted the DNA. The next thing we did was PCR. We use PCR master mix. It is a 2 times concentrated solution of Taq DNA polymerase, DNTPS, and all other components required for PCR except the DNA template and primers, which we got from our DNA extraction, and primers we use from DNA Q assay. This premixed formulated saved time and reduced contamination due to reduced number of pipetting steps required for PCR setup. This PCR mix then optimized efficient and reproducible PCR. The next step after PCR we did was gel electrophoresis. So we determined the approximate length of a DNA fragment of our cultures by running it in an agarose gel alongside a DNA ladder. And then the last thing we did was Kbase. It is a genomic annotation site. We used it to identify exactly what bacteria was present in the Boyce Park location, and we used it to identify. The next thing are the results. So my first one was CB2. It is identified as Elizabeth Kinglia Annotation. This bacteria can harm humans with weakened immune systems and young children and newborns. In the area where the drainage occurs, this bacterium causes bacterial meningitis in young children and adults with weakened immune systems, of course, and this could potentially lead to fatalities in this area. It was in the acid mine drainage, more likely due to mosquitoes that are present in the area. They carry thousands of bacteria from different areas that can bring it into the drainage system. Next was Abby's results, and she used AC1, which is identified as Luteobacterium species 9133. This is a close association with plants, so bacterial fungi complex that modulates diverse fungal phenotypes, which includes the potential for some fungi to act as plant growth promoters. So the reason for it was probably present in the acid mine drainage is it's present in soils since it helps aid in plant and / fungal growth in that area.
So the purpose of this research was for us to study the components of water collected from Boyce Park in order to really purify it down and identify its components. Based on the analysis that we concluded throughout the course of our research, our genomic assemblies contain hundreds of thousands of different sequences that we were able to analyze down to a single sequence through the use of all these different methods that we used, and then finally Kbase, which was our genomic annotation site, and that was how we were able to identify our prokaryotic genome. Further research into these specialties about these genomes could lead to a treatment plan that is designed to clean and purify the waters with these bacteria in it. It's definitely very important that we continue to study abandoned acid mine drainage because it can have such harmful impacts on humans, animals, and plants. So the amount that this research could benefit different life forms is definitely worth exploring. Yeah.
One second.